Started. Welcome, everybody. Welcome to NANOOS's webinar today. My name's Jan Newton. I'm the Executive Director of NANOOS, the Northwest Association of Networked Ocean Observing Systems. We are the ocean observing system for the Pacific Northwest, part of the United States IUS. And it is our endeavor to basically translate the ocean, to make ocean data from observations and models accessible, but also relevant. And a big part of making it relevant is making sure that we're engaging with the people who are using our products and get your feedback. So we're really excited today to have this webinar. We'll have a talk, and then we'll have time for question and answer at the end. We want to make this interactive. We're thrilled today to have with us Dr. Parker MacCready to talk with us about the LiveOcean model. In addition to observations, NANOOS hosts many models online, and LiveOcean is one of those products. We're really excited to hear from Parker about what you can learn from the LiveOcean model. Okay, Parker, over to you.
Thanks very much, Jan. So I will launch into my slides. So I'm Parker MacCready. I'm a research professor in the School of Oceanography at University of Washington. And I'll be giving you a bunch of information about the LiveOcean model, a little tour of what it does and how it does it, and also trying to focus on sort of evaluating how you might use it in your own research, or things you can, what can you trust more and trust less. There's a lot of people on my team who helped build the model. Specifically, I wanted to mention my postdocs, Jillian Xiong and Kate Hewitt, and grad students, Erin Broach, Aurora Leeson, and Dakota Mascarenas. And together, we put together a lot of the aspects that lead to this model. And of course, there's a lot of funding agencies that help, especially NANOOS and the Washington Ocean Acidification Center. Outline of the talk is shown here. I'll tell you what is LiveOcean, how it's created, where you can find the model output, and then a somewhat longer discussion of how reliable is the model for your use, going over some specifics, and then some new developments. You can see the model domain here on the right, and I'll give you some description of it in the next slide. So the model, it's like, think of it as a weather forecast, but for the ocean. And instead of focusing on things like, is it going to rain or how windy is it going to be, it focuses on temperature and salinity, but also the other things that we care about that make the ocean special, like phytoplankton and oxygen levels and ocean acidification. The model domain can be seen on the right. It covers the entire coastal waters and slope and abyss for Oregon, Washington, and some of British Columbia, going all the way around Vancouver Island. And then especially it also includes the coastal estuaries like Willapa Bay and Grays Harbor and the Columbia River, and of course the inland waters of the Salish Sea, Puget Sound, Strait of Georgia, and Strait of Juan de Fuca. The overlay here is surface temperature from one of the model outputs. So that's not data, that's model output. It's not from a satellite, it's simulated by a model. We do a lot of things that I'll describe in a second to make the model as realistic as possible. And of course, it simulates everything in 3 dimensions: currents, tides, temperature, salinity, and all the biogeochemical models that I'll show you some examples of. In addition, it makes, just like a weather forecast model, it makes a short-term forecast every day, in our case, a 3-day forecast. And that's the sort of the very front end of a long, at this point, about a 12-year hindcast that allows us to answer different types of scientific questions with the model. The big picture of how the model is put together is, first, on the left hand, we have to collect a bunch of different pieces of environmental information that are like the boundary conditions of the model. So there's the thing called, here, if you can see my pointer, WRF is a weather forecast model run at the University of Washington that gives us things like winds and surface heating and sunlight and so on. And then also we need information about the state of the ocean outside the model domain. So to give us boundary condition information that comes from a global model called HICOM. Then there's a lot of rivers and actually now wastewater treatment plants in the model. I'll be showing you a little more information on that. Can you hear me okay still? I heard someone. Anyway. And then,
yes, we can hear you loud and clear, Parker.
That's great. Thank you, Jan. And then it also has tides. Oops, get back there. So all those are assembled every day from different external websites and then used as forcing conditions for the model. The model itself is run in a University of Washington supercomputer called HIAC. And then the model output is post-processed and goes to a number of different places. And I'll show you examples of those. Most notably is the NANOOS NVS, or the NANOOS Visualization System, which is the primary place you would find LiveOcean output. But there's also a sort of custom LiveOcean website that I'll show you as well. So how can you find model output? So one place to start, if you don't get directly to the NANOOS NVS, is just to start at the LiveOcean website. And the best way to get there, the quickest way to get there, is just to search for the word LiveOcean with no spaces in your favorite search engine. And that should get you to a page that looks like this. The main things to be aware of are that there's a number of buttons that get you to different pages in the LiveOcean website. The main one up here, the NANOOS NVS, gets you to the NANOOS site, but there's other ones that, for example, daily forecast movies, high-resolution submodels. I'll show an example of a couple of those. These interactive tools that allow you to interact like a tiny video game kind of with both observations, model observation comparisons, and drifters that are calculated from the model. Then everything on the right-hand side is like deeper information, background. Sometimes these are things that are appropriate. You might want to use them in teaching for classes or just for learning about things yourself, like about how tides work or looking at annual cycles of salinity and oxygen and so on. Next slide. So if I click that first button, the NANOOS one, I get to then NANOOS NVS portal. And as I said, this is the main place to find LiveOcean model output. It serves a whole host of different model fields as map overlays that you can zoom in on and so on. And so there's things like oxygen and temperature and salinity. And it also has the capability of putting the model output that is the most recent couple of 3-day forecasts in the context of real-time observations. And I'm showing an example of that where I've brought up the model temperature field. You see it's for the surface. You could also look at different depth layers or even down at the very bottom. That's useful for oxygen, for example. And then over here on the right, there's a tab called if I click on one of these buoys, it brings up a thing called the comparator. And if I click on that, it'll actually allow me to look at a comparison between recent temperature observations in this case and model output. And I just want to show off some new capabilities that have recently shown up in the NANOOS NVS. And for that, I'm going to go to the NVS page as get away from there. Okay. So I'm hoping that what you can see now is the NANOOS NVS, and I have the LiveOcean water temperature brought up. You can see there's this thing called layers. And if I click on layers, I get a bunch of different choices, different models, different atmospheric models. And then for LiveOcean, you can see there's 7 different variables that you can click on, like salinity or temperature. Then when you have them, you can see a really great new feature in NANOOS is that as I drag my cursor around the model field, it shows you in what the temperature is at that location. So for example, if I was looking at oxygen at the bottom, there you can see, and I actually want to change the color bar, fit range to data. And you can see that there's also new capabilities with the color bar. You can choose your own color map and the ranges. Really nice, beautiful work that they're doing. And now I can see what the bottom oxygen concentration is here in different places. And of course, you can zoom in and look at it in much more detail. So that's that. Stop share. Sorry, it's kind of a 3-ring circus here. Share. Back to the Chrome PowerPoint. So those are 2 main places to look for model output. And then the question that I really wanted to address is how reliable is the model for your use? And of course, the answers are different for different properties and locations. So I'll just go through a few examples. What if I care about temperature or marine heat waves? And I wanted to highlight the fact that the model now has a much longer continuous hindcast, about 12 years now in total. And the plot that's shown on the right is monthly mean surface temperature. You can see the columns are different months, and then therefore 10 years in this case from 2014 to 2023. And what I've plotted is actually the monthly temperature anomaly. So it means I've taken the average January, for example, of all those 10 years and subtracted it from the January that I see in 2014 or 2015. With a plot like this, you can see anomalies and especially marine heat waves. So the big one that came ashore in late 2014 lingered in 2015, especially in the winters, reappeared in the sort of spring of 2016. And there's a few other events like 2019 as well. The model has, in general, good skill with temperature, with errors about 1 degree centigrade. And you can see that more quantitatively this is the type of plot that we use in the coastal modeling group to evaluate model skill for different purposes, or this is one of the tools that we use. So each of these is a comparison of observed fields. So that's the x-axis is observed and the y-axis is the modeled value. And then each panel is a different variable. CT is something called conservative temperature. It's the same as potential temperature, but really just think of it as degrees C. And then the green line shows a 1-to-1 line. So if all the dots, which are thousands and thousands of observations at the locations shown in the map from many different sources that we've collected, if all of them were exactly perfect in the model, they would all lie along the green line. And you can see that there's some scatter around the green line. The blue dots are for the deeper bottles, and the red are for the top 10 meters. You can see there's more scatter near the surface, less scatter at depth. Then in general, for physical properties like salinity, that SA, and temperature, we do better. For things that have biological influence like oxygen or nitrate and carbon variables like DIC or alkalinity, there's more scatter. That's just typical of how biogeochemical models function these days. I also wanted to bring to your attention a new interactive tool that allows you to explore that exact same kind of data yourself. So this is accessible from the LiveOcean website in the category of interactive tools. And you can see it's the one down here called Model versus Observations Viewer. That brings up a page that looks like this, which I just want to demonstrate very quickly here. I can figure out how to stop share, Chrome, share. So I'm in LiveOcean. I go to the Model versus Observations Viewer, and it brings up some of those property plots like I just showed you and a map, but now it's interactive. I can choose which year I want to look at. I can choose which month I want to look at and a region. So I could select just the bottles here in this region off of the mouth of the Strait of Juan de Fuca. They show up dark blue. And then as I drag the time slider across, you can see different months light up as red on the property plot. There's also a similar tool, but just focused on observations. So same thing, but now I'm only showing observed values versus depth of things like temperature, salinity, oxygen. And again, you can drag across. So this is just a way to learn about and appreciate the multitude of amazing observations that are made by different groups in our region, like Department of Ecology, Environment Canada, WOAC, the NOAA, and several other organizations. Stop share. Share. Sorry for the back and forth. PowerPoint. Okay, so back to my slides. I think that's all the back and forth I'm doing. So temperature, as I said, pretty good. What if I care about bottom hypoxia on the shelf? So this is something that might be relevant to a somebody looking who's fishing for Dungeness crab on the shelf, for example. In general, on the shelf, bottom oxygen is reasonably well represented. That's a non-quantitative way of saying it. We're biased low, about 0.5 milligram per liter in general. One of the movies that's on the website is the one I'm showing here that shows the last 5 days of bottom oxygen. Of course, you can also look at the bottom oxygen fields in the NVS portal. We use other things besides those bottles to also evaluate the oxygen on the shelf. In this case, the Olympic Coast National Marine Sanctuary moorings. And I'm showing 8-year time series of oxygen at 2 different depths. It is near the surface is red, near the bottom is blue. And so you see, of course, especially in the summers in our region, bottom oxygen goes to sort of hypoxic or sometimes almost anoxic values. In this plot, the fainter or transparent line is the model field, and then the darker line is the observations, which those moorings are only out for the summer months, and so that's why they're limited in time. In general, we do pretty well for representing bottom oxygen here, and these results are similar for all 10 of the Olympic Coast National Marine Sanctuary moorings. That's a great asset we have here in our region. Turning to another thing you might care about is the effects of treated sewage in Puget Sound, the nutrient loading and potential for eutrophication and hypoxia caused by that nutrient loading. The model has been substantially improved in the last couple of years by adding many small rivers and wastewater treatment plants, all data developed with a lot of effort by our Department of Ecology. And those are now, there's about 279 rivers, big rivers and small rivers in the model, and wastewater treatment plants. In general, we do not as good inside the Salar Sea with oxygen. In this version of the model, the bottom oxygen is biased low, in general by about 1 milligram per liter, and up to the hardest place for us to model right now is Hood Canal, where it's biased low by about 2 milligrams per liter. So we believe that we overpredict hypoxia at this point, although there's still, well, let me show you a map of that. Here's some work being done by our graduate student, Aurora Leeson, and she is looking specifically at the effect of sewage treatment plants. So to do that, we do 2 nearly identical model runs, so the same physics, the same rivers, winds, everything, tides, and we just turn off the nutrient loading from wastewater treatment plants. In the map in the middle of the screen here, you just see oxygen values in milligrams per liter averaged over the summer months or sort of fall months when hypoxia is the worst. So red is hypoxia. You see that showing up in South Hood Canal, as we expect, and a few other places that are known problems. And then in the right is the different of the run, the sort of the standard run that has anthropogenic forcing like wastewater treatment plant nutrients, and a run without that. And if it's red, that means the oxygen dropped, and you can see the color scale. It dropped by sort of 0.1 to 0.2 milligrams per liter, something noticeable. I'll show you something about how, well, anyway, this is an active research, so don't take this as a finished result at this point. This is, yeah, just exploratory research. Really, it's trying to reproduce or check on the calculations that Department of Ecology is doing with a different model to make decisions about wastewater treatment plants. As I mentioned, we think that we're biased low in oxygen, and especially Hood Canal is a problem for us. And so we are always trying to improve the model, and improving oxygen in Hood Canal is one of our main targets for the next version of the model. We roll out new versions of the model about every year and try to make substantial improvements each time. And we do this in a data-driven way, meaning that there's many things we could change in the model, parts of the biogeochemical submodel, for example, parts of the circulation, anything that we can prove, but we try to have a data-driven reason to make the changes we do. In our case, one of the main things that we're using is a new paper from Santana and Schul. David Schul is a faculty at Western Washington University, who did a bunch of sediment flux measurements in Puget Sound. And we found that our sediment oxygen demand was noticeably higher than what they measured. And so to change this, we started burying particulate organic matter that came to the bottom. I know this is a lot of jargony stuff, but I hope that it's meaningful to some part of the audience. And we have other reasons to believe that there actually is more burial of organic matter in Puget Sound or the Salar Sea than there is on the shelf. And so when we run the model with allowing for more burial, we see that the comparison of model to observed oxygen goes from what's in this panel, current model, to this version on the right, where we've mostly removed the oxygen bias and our overprediction of hypoxia, which was happening down here. Another thing you might care about is aragonite saturation state in Willapa Bay, of concern for oyster growers, because if it's too low, then larval oysters don't survive. The LiveOcean website shows a forecast movie of, in these high resolution submodels category, of Willapa Bay and Grays Harbor surface and bottom, what we're calling ocean acidification, but it's really aragonite saturation state. And then the model movie looks like this thing that's moving on the right. This is a high resolution nested submodel. big LiveOcean has about 500-meter grid in this region, but that's not really good enough to work well in Willapa Bay and Grays Harbor. So we have a 200-meter nested submodel. And you can see it has wetting and drying. That's where it goes, these white bits that appear at low tide. And that's what's being shown here. So that's one place to access that. We are working on having this nested field appear in the NVS overlays in NANOOS, and we're getting close to that being done. With respect to how good is that aragonite saturation state, if I just compare, in this case, my model data comparison plot for omega, which is aragonite saturation state observed versus modeled, you can see that we're biased low. That is, the dots are low by about 0.25, which is you'd like to do somewhat better than that. And there's also a lot of scatter. That's to be expected. This is another thing we're trying to improve by working on the carbon model that works on DIC and alkalinity. In order to do this specifically for Willapa Bay and Grays Harbor, we need to start doing, for one, model data comparisons of moorings that are maintained there. And that's a target for work in the coming year. If I care about harmful algal blooms that affect coastal razor clams, I think at this point, the state of the science is that in our region, models are not able to predict when pseudonychia blooms will occur compared to the other diatom and other phytoplankton blooms that happen in our region. And moreover, we're not able to predict when those will become toxic. And that's an aspect of active research. We're making some progress on that, but not yet. So but the managers that open and close the razor clam harvest do get a lot of scientific information. A lot of it's observational, and it's packaged in something called the Pacific Northwest Harmful Algal Blooms Bulletin. And you can see it has a bunch of observations on page 2 of that bulletin. You can see the LiveOcean model is just one part of that system that is information for the managers. And what we do is a custom particle tracking experiment every day. And this is a movie that's available on the LiveOcean website that shows where do particles that originate in 2 known HAB hotspots, that is Hecata Bank to the south and Juan de Fuca Eddy to the north, where are water from those places going to go in the next 3 days? And that's very much dependent on the state of the winds. And then I want to finish up, look, I got through really quickly, with another new capability of the model, which is that we have a second nested submodel in addition to the Willapa Bay and Grays Harbor, that's run as part of the daily forecast. You can access this through the LiveOcean webpage, again, in the category of high-resolution submodels. And it's these last 2 buttons, South Puget Sound surface temperature, South Puget Sound salinity. And this is, the movie shows surface temperature in South Sound. So this is a 100-meter grid, covers the regions of South Main Basin, Tacoma, Carr Inlet, Case Inlet, and Bud Inlet. It's especially interesting to me because I live down here, and so I like swimming in the water right around Dana Passage, and I want to know how cold it's going to be. This submodel also does all those same biogeochemical fields, and so this is something new that's just been rolled out in the last week or so, but it appears daily as a forecast on the LiveOcean website, and hopefully sometime soon on the NVS site. And that is all of my presentation, so I am happy to answer questions.
Wow, thank you so much, Parker. That was a fascinating talk, and I just want to say the answer to your question is it's going to be cold when you swim.
Ah! No, but what about all that orange water?
I know, and I was looking at it. There is, yeah, but
It
your
look
max
warm.
scale is not much over 10. So hence my reply. Thank you so much for that input. So this is the point where we welcome questions. And so you have two options. You can raise your hand or you can write them in the chat. And I'm going to read a question from Herb Munson that says, how do we get numbers to deal with as well as image depiction of those numbers? So I think you're seeing the images. And I think Herb's asking, is there a way to actually get the numbers?
That is a great question. And there's a few answers to it. There isn't like a standard, like a thread server or something for the LiveOcean output at this point. And it really, the answer to your question really depends on what numbers you want. The difficulty with serving model output is that it's too large. That last decade, Hindcast is about 100 terabytes of three-dimensional hourly model output that we save. And so that itself is not movable. I do often make custom extractions for people of like simulated mooring locations or the entire tidally averaged surface field for a year or things like that. Or that's the kind of thing we do. And then also it is possible to access the files that NANOOS gets of their extractions of the model for the last, there's essentially a bunch of variables every 4 hours for the last 3 days appear as a single netcdf file, and that's extracted at the surface, 3 different depths, and the bottom. And those actually are publicly available, although we don't at this point advertise the URL for them. But if you want to know, if you want something like that, let me know. I wish there was a sort of a one-size-fits-all answer, but I've handled many, many, many data, or model output requests in the last 10 years, and everybody wants something different. So that's where that is.
Yeah, thank you, Parker. And I can attest, I co-teach a course up in Friday Harbor, the University of Washington's Marine Lab up there. And increasingly students are saying, could we get this output for this point? And Parker comes through. So really want to thank you, Parker, for that. Okay.
I really want to automate that. But
yes, I know. Yeah. So I don't see any hands up, but I'm going to read some more comments and questions. Lynn Wenberg says, this is fantastic. My group uses NANOOS for improving timing of local bulk help surveys during the summer. It's great to hear. She has 2 questions, the first of which I can answer. Will this presentation be made available to attendees? Yes, we are recording this and it will show up on our website. NANOOS has now a space for videos, and the webinars that we host are going to be displayed there. So it might take a minute to get it there, but it should be there very soon. And then the second question is to you, Parker. Are you available to make this presentation to a larger audience, such as the Northwest Straits Commission and 7 Marine Resources Committees?
Yeah, I'd be happy to.
Yeah, and I just want to really reiterate, the whole idea behind NANOOS is really to take the best of academic or federal research and to combine it with the needs of the people in the region. And that's what you're really seeing in this instance. I've seen Parker in action, listening to shellfish growers, listening to people working on habitat restoration, and really so many of the improvements you see from LiveOcean are because of those conversations. So a shout out to Parker, as well as the way that IUS is conceived to really bridge that between institutional barriers. Okay, here's another one from Natalie Coleman. Great developments, clarifying questions since ecology started taking surface samples to calculate omega in January of 2022 at Willapa Bay and Grace Harbor within the bays. Is your model pulling any data from within those bays, or is it relying mostly on moorings? Sampling the coast is hard, which I will agree with. Over to you, Parker.
Yeah, good question. As far as I know, we are not yet using the carbon samples from ecology in Willapa Bay and Grace Harbor, but we do routinely use their monthly, they do monthly CTD casts at several stations in Willapa Bay and Grace Harbor. So I invite you to actually go to the LiveOcean website, go to that model observations viewer or just the observations viewer, and you'll see there's little dots on the map in Willapa Bay and Grace Harbor. All those little dots are ecology stations. And we do routinely ingest those. And we make a lot of use of ecology data and make every effort to keep it up to date and processed in a way that we can combine with all the other data sets we use. So you can, yeah, give it a try. You can use that observations viewer and see the data yourself, or how the model does in Willapa Bay and Grace Harbor.
Yeah, that's a very cool new feature, Parker. Next question is going to be a live question from a dear friend, Richard Dewey up in Canada. Hey, Richard.
Hi there, Jan. Hi there, Parker. Great presentation. And
Hi, Richard. Good to see you.
Yeah, good to see you too. Just fabulous work. This is really, really impressive, and it's a huge legacy for everyone. It's a massive effort. I just wanted you sort of brushed over it, but some of the plots you had there, certain biological elements or biogeochemical elements are struggling, and one that popped right out was the chlorophyll. Do you have any comment about why you're tracking some of the others really quite well, which are related, but chlorophyll seems to be a real challenge?
Yeah, that's a great question. I think partly it's chlorophyll itself. Like, what we keep track of in the model is nitrate in phytoplankton, or sort of, we use nitrate as a currency in the biogeochemical model, and phytoplankton is just one of those places where nitrate, can move into or move out of. And then converting that to chlorophyll it's really quite uncertain in the observations. We take our model field and multiply it by 2.5 to get to the units conversion, but there's really wide error bars on that. The other reason, I think, we know that regardless of the conversion, there's ways in which the model phytoplankton blooms are different than real phytoplankton blooms. And we've done a lot of comparisons with satellite color, for example, to know this. So additional to those chlorophyll measurements. And what it shows is that the real blooms are like bigger and peakier. That is, they go up faster and die faster. And I think it's because in the real world, there's lots of different types of phytoplankton, and they're each one optimized to a different condition. And so somebody's going to be a winner, and the model doesn't have all those different types of phytoplankton. It just has one. And so it's kind of representing the average.
Yeah, so no, I think that's exactly what I anticipated, that it's a species mix problem.
Yeah.
And yeah, okay, that's great. Good, thank you.
Yeah, I'm going to chime in a little. Oh, sorry, Parker, continue.
No, go ahead, please. Tell me what.
Well, as a biological oceanographer here in this conversation, I can say, if you take water samples for chlorophyll and you have a mooring with a fluorometer, and then you take phytoplankton abundance samples, those three data streams are never going to look like each other by and large. There's so much that goes on just with quenching and other things with the sensors and how the sunlight influences that. Discrete, from the desktop analysis or benchtop analysis is kind of like the holy grail. But then trying to convert it to phytoplankton biomass, as you guys were identifying, it's really tough. And then throw in satellites and my mind explodes. But I think all of them are useful because they're giving us insights into something that is actually very hard to measure. Yeah.
Thanks.
Okay. I don't see more hands up, so I'm going back to the chat. Kim Martini says, thanks, Parker. This is great. Besides your cold swims, are there other people or groups using the 3-day forecasts?
The people that use them, I sort of alluded to them in the talk. So one is the people doing the sort of razor clam management on the coast. I believe that the Dungeness crab fishers look at the bottom oxygen on the shelf. I think it's still a work in progress to make the aragonite saturation state be something useful for shellfish growers, but again, we're working on that. I give talks at shellfish grower conferences and continuing to keep that dialogue open. I think one problem is, what are you going to do about it? And just knowing about it, that helps somewhat. But there's also other groups, like there's a company called Scoot that it's a private company that gives data services for aquaculture, and they ingest the model fields and make use of them. Another user is Susan Allen's group, runs a daily forecast model for the whole Salish Sea, and they use the LiveOcean fields for the open boundary condition at the mouth of the Strait of Juan de Fuca for their model. Yeah.
Yeah, I wanted to Parker's horn a little bit. He and I wrote a proposal several years ago to institutionalize LiveOcean more, and we were able to get over 20 letters of testimonials of people saying that they're using it. And in the chat, Simeon Baldwin says, Kim, yes, I, as a U.S. Coast Guard auxiliary boat crew patrol mission planner, use the 3-day surface temperature forecast for determining our PPE clothing requirement for an upcoming patrol. Under 50 degrees surface and under 50 degrees air, we are required to wear full dry suits, etc. So that's great, Simeon. Thank you for letting us know. And it's wonderful to hear these uses that go beyond even what we know. Okay. Derek Kaufman says, so impressive. Can you discuss more where data comes from? How much of the output is calculated or interpolated? How are those calculations created and adjusted?
Excellent question. So the model itself is 100% a simulation. So you could think of it as not data at all. It's 3-dimensional fields and we have equations for how those things change over time as they're influenced by, for example, the phytoplankton concentration is influenced by how sunny it is and how much nitrate and ammonium is available. So those are all simulated. However, the forcing fields, like how windy it is, which really pushes river plumes around, for example, or what the water properties coming in from the open ocean boundaries, those come from other models. But those models, like the weather model and the global ocean model that we nest inside of, they use a lot of what's called data assimilation, meaning they're constantly adjusting their fields to be closer to reality. That's why a weather forecast model keeps looking like the weather, is because they keep nudging it to, restart every day with something that looked like the right initial condition. The same is true for our rivers. They're constantly updated with real-time observations or short-term forecasts. Other than that, we don't adjust the model fields. But I will say that in coastal waters in general, these limited domains, they're so strongly affected by the boundary conditions that they tend to, respond to them. And that's why the model keeps looking like observations to the extent that it does, is because the boundary conditions are basically correct. I hope that answered your question. I know it's a funny concept to think that it's all just simulated, but I will say that under the hood, the weather forecasts that you look at are doing exactly the same thing. They are a three-dimensional computer simulation that uses some environmental information and then the laws of physics to drive them forward in time.
That's a really good question or answer, Parker. I think it's important because we're so familiar with weather forecasts and maybe not so much with what we're talking about here. Scott, I see you have your picture on. Did you want to ask a question?
Yeah, I'd love to. Thank you,
Great.
Parker.
Hey, Scott.
It's wonderful to hear you talk about this wonderful model that I think many of us have enjoyed for quite a while. And it's great to see it evolving with your postdocs. As a quick aside, as a sailor here in Puget Sound, I'll say that there's been a lot of attention in my world to the surface current and various tools for predicting surface currents on finer and finer scales. So that's another 3-day forecast user that I think is evolving. A bunch of youth sailors know about LiveOcean now, and they, for better or worse, they think about whether it's useful for their racing. But the question I wanted to ask, Parker, I think harkens back to Neil Bannis's work. And my favorite part of LiveOcean is to go watch the year-long animation of the phytoplankton. I think it's on the far right side of your LiveOcean menu. It was great to see a 3-day forecast for chlorophyll show up in NANOOS. So I've been looking at that and trying to use it to contextualize more and more local observations of marine mammals and other parts of the ecosystem, at least in the central main basin. So I just wanted to flag that I think that's an area that's useful already, but I'm curious to know, what do you see in terms of quantifying skill for phytoplankton, let's say in the next 5 years, or opportunities to even think about a zooplankton trophic level, let's call it. And I'm particularly interested in the possible interplay of improved monitoring efforts, at least here in Puget Sound proper, through the PSEMP vital signs. The Puget Sound Partnership's vital signs include now some revised zooplankton indicators, which I think are at least seasonal. And it might be interesting to juxtapose those sort of aggregated monitoring efforts with a zooplankton, let's call it a prediction or a layer in maybe subregions of Puget Sound. You think that's possible, or is there other sources of empirical data that could be used to sort of advance the model up into the next trophic level?
Good question, Scott. So the model does have zooplankton in it right now, and that's not shown as one of the fields in NANOOS, but we do have it there. And I think kind of like chlorophyll, but worse. I think I have less confidence in our ability to simulate zooplankton because the difference of different species is even, more extreme. Some swim up and down, some, they do all sorts of things. And the zooplankton parameters in our model right now are really optimized in one of these data-driven model development things, but really for the coast. And that was work that Neil Bannis was doing back when we had observational projects on the coast in what's called the RISE project for the Columbia River plume. And I think it would, I think we could be doing much better than that inside the Salish Sea. The big source of data allowing that is this long time series of zooplankton observations pioneered originally by Julie Keister and now being carried on by the new biological oceanography faculty at UW, Karen Chan. And that's a tremendous data set. If you look at it, you'll appreciate how noisy zooplankton is and the different species that show up in different places. But I think connecting to that will be an important future step. The other thing I would note is that, apart from bottom oxygen being, having some biases in Puget Sound, for example, that we're dealing with sediment, our treatment of sediment, I think the surface field, the nitrate shows up too high. And I think working on phytoplankton and eventually zooplankton will be the next target improvement after bottom oxygen. So getting surface fields more right. We're not using up enough nitrate in the surface water at this point. It's like me rambling about the things in my head. I'm not sure if I answered your question, but thanks.
Well, I think it would, go ahead, Scott.
Oh, that was a great answer, Parker. And the other thing I'll flag for you all is I've been very impressed with the crab larval monitoring and how quickly it seems to be growing. So then another thing that I think might provide like more spatial and temporal resolution going forward is those light traps.
Good point. And the crab, I especially care about the crab in South Puget Sound because our season is like permanently closed at this point for Dungeness crab. And, that's a kind of thing that's a science project that's waiting for the right funding to come along. We've tried to get funding for that in the past and it hasn't worked, but I suspect it'll happen in the future. Thanks.
Yeah, that's exciting. Karen Chan is working with the folks with the light traps and that network. And so looking at the zooplankton layer in LiveOcean and kind of taking a deeper dive would be really interesting to start branching into that trophic level. Okay, gang, we have 7 more minutes. Would anybody like to ask a live question? If you want to, go ahead and turn your monitor on, raise your hand. I don't see anything in the chat. I do want to flag that there are different models that NANOOS hosts, and what you've seen is one example, LiveOcean. And if you go onto our NANOOS website and the Data Explorer, NVS, and Rachel, if you could put that in the chat, just the direct link, there's a landing page that has all sorts of different models. If you go to layers, and you will see models from University of British Columbia, for instance, Oregon State University, the CMOP model that's focused on the Columbia River. Each one of these has a different specialty, a different place that it's really focused on. And I think the great thing is being able to compare these models. And so that's great. All right, I am not seeing any further questions. One thing I want to do is, first of all, thank Parker so much. Thank you, Parker. That was a great talk.
Thank you for organizing.
Yes, and the other thing is to say to this crowd, we have over 70 people online. We had about double that register. Please stay in touch with NANOOS. Send us your questions, your suggestions. If you just go to NANOOS and you click contact, that sends an automatic email to several of us here at NANOOS. If you have ideas for additional webinars, we're looking to do some more, hopefully on the Tsunami app and the Fishers app. So anyway, don't be strangers, stay in touch. Let us know how we can continue to improve NANOOS, and just again, thank you so much, Parker, for all your excellent work on this. And thank you everybody who tuned in. This is recorded and you will find the recording on the NANOOS website. And so I think that's it for now. Thanks everyone. Bye.